So what I'll talk about today is the work that led to the Lasker Award, but also I'll go beyond that. I'll talk about the very process by which insights into how microbes, single-celled organisms work, can actually give rise to insights into brains, into how the mysterious properties of our own brains can arise from the properties of their components, all the little cells that constitute them. And that's an amazing story, how algae, single-celled plants, and ancient forms of bacteria can help us understand ourselves. And as uplifting as the story is, of course, it's always framed by the backdrop of the suffering that is present in the world and what we need to do to address that suffering. As you heard, I'm a psychiatrist. I focus on depression and on autism spectrum disorders. And there is, from where I sit, no more mysterious, challenging, and frustrating disorder than depression. This painting, which some of you might know, is so evocative for me when I think about my patients, the people I know with depression. It's not just the anguish, but also I think this painting captures the isolation of depression. This is somebody who can't communicate what's going on inside. It's something not understandable to friends, to family, which creates misunderstanding, which compounds the disorder, the disease, and the suffering. And one hope is that science will help us, that we'll at least come to an understanding. Maybe we can get to a point where this opaque organ, so mysterious and complex, becomes accessible to understanding, so that we could end up reading it like a book, to see how the connections, the projections from one part of the brain to another carry information, cause things to happen, give rise to or ameliorate symptoms, and how the properties of the brain arise from its parts and its connections. And the story that I'll tell you about is that there's hope, that there is progress in determining what actually matters in the brain, what the parts do, and how they work together. And the nature of this story and the Lasker Basic Medical Research Award is, I think, an uplifting and intriguing story that brings promise for the future. Now, I can't even discuss the Lasker Award without honoring my two co-recipients, Peter Hagemann and Dieter Osterhelt. These are amazing scientists from Europe who did foundational work. We were collaborators, friends, colleagues, and I'll tell you more about what they did. But it turns out we have to go even farther back in time to 1866 to truly understand the roots of this transformational advance in science and in medicine. Andrei Fomentsyn was a botanist in Russia, in St. Petersburg, and he collected freshwater algae from a river near his laboratory. And he noted that algae in a dish would move toward the side of the dish where light was present. So this was plant behavior, single cells swimming toward light. Turns out the algae, these single-celled algae, have flagella, so they can swim through the water. Unlike most plants, they actually exhibit real-time behavior. But they do photosynthesis like all plants, and they have chloroplasts, but they also have a little eye spot, a little spot that helps them detect light. And with brighter light, the algae would back off to an intermediate location. So this was not a trivial behavior. It actually had some optimizations and some complexities. So this turns out, as we now know, to be mediated by a beautiful class of proteins, single biomolecules called microbial rhodopsins. But this was the first evidence that they existed. It took a number of years, more than 100 years, to sort out how that worked. Dieter Osterhelt found microbial opsins from archaebacteria. Peter Hagemann found a special form from the single-celled algae. In my own group, we identified from this colonial algae called Volvox, a special form that allowed us to get to single cell control in the brain. And together, what we were able to do was develop an understanding of these beautiful natural proteins. Now here is Dieter Osterheld and his son Patrick, and myself and Peter Hagemann. All these pictures are from many years ago. We're all slightly damp, it turns out, in these pictures. This is not intentional, but just how it turned out. This picture is of Dieter Osterheld in just near the coast in San Francisco. He was on a sabbatical from Munich and was working at UCSF. And this is at the time of his transformational discovery. Likewise, Peter and I were caught in a rainstorm in England, and we ducked into a little restaurant to get some food. And this was in the midst of one of our very exciting collaborations. So what did Dieter do? What he did, and this emphasizes, I think, very clearly how important these deep roots in basic science are for all three of us. He was studying the bacteria. He had no thought for understanding psychiatry or even neuroscience or the brain. He was very curious about microbes and did some brilliant work. He noticed quite unexpectedly that a class of archaebacteria, an ancient form of bacteria, would change color in the right solvent with the right light exposure. It would change from purple to yellow. By the way, Jeff Friedman, who did a secret science talk not long ago, generated some very nice images of this for the Lasker Award, which I borrow here. And what Dieter next found was that this caused a pH change. It caused a change in the acidity of the medium, which was even more mysterious. And he did some work on this, and it turned out that there's a protein not known until that moment called bacteriorhodopsin, which in response to light moves hydrogen ions, protons, across the membrane of cells. And this changes acidity. This is, in a fundamental way, acidity. And by acidifying the extracellular environment, that drove production of energy, production of ATP, which is the cell's currency for energy, as the protons then rush down the new concentration gradient. So beautiful basic science, an organism that makes a beautiful protein, a single light-activated biomolecule that receives a photon and changes the acidity and does that to make energy. What a very cool discovery. What did Peter do? Well, what Peter did was some years later, he was studying those single-celled algae, and he found that when he could catch one and hold it on a pipette, that there were some currents that would flow in response to light, and they were very fast. It only took about 10 milliseconds for a current to flow from the eye spot to the flagella. And he was curious about this. He thought that meant that there might be a light-activated channel, not just something like bacteriorhodopsin, which moves one ion per photon, one particle of electricity effectively for one particle of light, but something even more powerful that let many ions across the membrane in response to one photon. And this was an unusual, even heretical idea, but he turned out to be right. And this class of microbial rhodopsins was called channel rhodopsins. And so related to these, just a little different in opening a channel instead of moving one ion across the membrane. So this was beautiful work. And I did a couple things to contribute to this. One thing that I did was understand the structure of these proteins, how they actually work, diving into the high resolution structure with measures like X-ray crystallography, which is, for example, how the double helix of DNA was identified. And what we can see once we look at the high resolution structures of these channel rhodopsins is that they are quite amazing proteins. We're zooming in to the angstrom level, 10 billionths of a meter. And we can see the pore going across the membrane through which the ions flow. We can see all the little atoms that together make the channel. And there are parts within this protein that absorb the light, that receive the photon. And it turns out there's a vitamin A-like molecule called all-trans retinal that absorbs the photon and develops a kink, and that causes the channel to open and lets ions flow across the membrane. Now, it turns out there are many of these channel rhodopsins. This is sort of like a tree of life that you might see when thinking about how different species and different families of the great tree of life evolve and derive from each other. Proteins do that too. They can even hop from one species to another, or their genes can hop from one species to another. And so you can make trees of proteins just as you can make trees of species. And the original channel rhodopsins that Peter and I identified are over here. These conduct cations, positively charged ions. Turns out there are others that pass anions, negatively charged ions. And we identified the structure of that one in 2018. The cation ones we found in 2012, got that structure. And then more recently, this family that's selective for certain kinds of positively charged ions. And these we call the pump-like channel rhodopsins, and that's a whole other family. We just reported that structure. The original ones that Dieter Osterheld identified are in this group. These are the pumps. And so we've got this family photograph, if you will, now of how what these different proteins are and how they work. And if you zoom in, you really get some insights, and those insights turn out to be useful. If we zoom in, this is a very close-up view, now really at the 10 billionths of a meter level. Imagine you're inside the layer of lipids that encases a cell and separates it from the outside. And this is a single channel rhodopsin molecule, all these green cylinders. It's got 7 of these cylinders that pass across the membrane. And these blue arrows are the channel. This is the path through which ions flow. All these gray spheres are water molecules to give you a sense of scale. These are water molecules that are on one side of the membrane of the cell or on the other side. And these are the ions that can pass. This is the pathway through which the ions can flow. This is that vitamin A-like molecule called all-trans-retinal, and it's encased in its own little pocket. And it's so tightly encased that when it develops a kink in response to the light, that allows the channel to open. And we sorted out a number of details of what all the atoms do in this structure. And this is a review that Peter and I wrote a few years back. And it's not just for understanding, although that is enough. We're curious about how these natural proteins work, and figuring that out is kind of a reward in itself. But also, we can make new kinds of function once we understand how they work. We were able to make very fast blips of electricity come out, going at hundreds of times a second with one particular modification of the atoms right here. And that was in response to blue light, these blue light pulses, as you can see here. We also could make red light driven pulses of activity through other modifications, including one up here. And then we could create more longer lasting forms of changes, things that would last for tens of seconds or even minutes. We even could create different kinds of ions flowing across the membrane. We could turn something that moved positively charged ions into one that made negatively charged ions flow across the membrane. And this proved that we understood how these worked pretty well, that we could make these changes guided by the structure. But also, these modifications turned out to be really, really useful for neuroscience. And how those became useful is shown here. And this is what we call optogenetics. Now, one challenge that's made the brain so hard to study is essentially every neuron, every brain cell is an electrical device. It responds to electricity, it creates electricity. And so if you've got a bunch of brain cells doing very different things that are right next to each other, and you put in an electrode, or even if you use a magnetic impulse, all the neurons more or less look the same, and you won't be able to selectively stimulate one or another. You'll get them all because they're all electrical. All neurons have electrically active channels that respond and cause spikes to fire, little blips of activity that are the currency of information flow in the brain. But you can't figure out how it works because all the cells respond the same. What optogenetics lets us do using these microbial opsins from these microbes, because these are single proteins encoded by single genes, little bits of DNA, we can put those bits of DNA into one kind of cell or another, and then use light instead of electricity. None of the cells respond to light normally, But the ones we do use genetic tricks to put in these light responsive proteins into, those become the ones that respond. And maybe we'll have one kind of channel rhodopsin that responds to blue light, and we'll put that into one kind of neuron, and maybe another one that responds to yellow light, and we'll put that in another kind of neuron. And now we can really start to control things with great precision. We can turn cells on. It turns out the positive ions coming in turns neurons on. Negative ions coming in turns neurons off. We can turn cells on or off with different colors of light and with genetic specificity and with high speed. So that is the nature of the opportunity, and that's what made this exciting. And over the course of about 15 years, we leveraged our basic science understanding that we built up on how these proteins work and developed ways to control even single neurons, elemental components of the brain, even within living, behaving mammals like mice. And this started about 15 years ago. I did a very simple experiment in 2004. This is a neuron growing in a dish, in a petri dish in the lab in plastic. And this yellowish green label is the channel rhodopsin that I put into the neuron. And this was a neuron that I didn't stimulate with light. This is one that I did, and you can see in the middle of it, in the nucleus of the cell, there's a red label, and that's a marker that the neuron was stimulated, that it was electrically activated by the light pulse. And I noticed that the cells that I stimulated with light had more markers of this activity than the ones that I had not. And that showed me right away that it was working, that I was able to take a gene from algae, microbial algae, single-celled algae, put that into mammalian neurons and control them with light. And so from that moment, a lot of interesting things happened. It took about 15 years to get to the single-cell control level in mammals. We had to use this channel rhodopsin from Volvox, this colonial algae, and some engineering around these atoms here to make a very robust red light activated channel rhodopsin. And I'll play you this movie here to show you how it works. So this is a mouse, and we're using light. We're playing in light of particular wavelengths that we know these cells respond to. And what you can see here, first of all, I'll pause the movie and tell you a little bit about this. Where you see a glow, this orange amber glow, that tells you that neuron is being active. And we've put in a little reporter that glows effectively. It changes its fluorescence when the neuron is active. So that gives us a readout, lets us see optically that the cell is responding. Now, this is a view into the brain of this mouse, and a bunch of these cells, individual cells, this is one cell, okay? It's about 25 millionths of a meter wide. Some of them, all the cells labeled T, T3, T5, T7, these are cells I'm targeting. So I'm calling them T, we're delivering light to those cells. Other cells, we're not targeting, and these are the NT cells, NT2 and T3. Okay, so now we'll play the movie. You can look at these two gray NT3, NT2 neurons and see they're not responding when all the other cells are being stimulated. They're doing their own thing. But all the targeted cells are being stimulated together with the light when it comes on. And you can see a summary of all that happening here. Here are all the targeted cells responding. The non-targeted cells are not responding, even though they're right next to the targeted cells. And so we're coming in with light, focused light beams, targeting individual cells that have a channel rhodopsin in them, and we're able to turn the cells on at times that we want, all within a living mouse brain. So this is really exciting, and this allowed us to do a lot of things. The overall picture of this is a long-held principle in biology. We like to do what we call gain of function and loss of function. We like to understand how something works to see if something's important. We like to give it, and we like to take it away. And that tells us a lot about if this process, this thing, this component, this element, this gene is important and what it's important for. And Francis Crick of double helix fame also had thought about this, and he thought, you know, for neuroscience we need, and he described this in 1999, he said we need a way to control individual kinds of cells. And he even thought maybe light would be a good way of doing it. He had no idea how to do it. In fact, he even said this would be far-fetched, which is quite a statement coming from him, but he thought that this was important. And this was this traditional genetic biochemical thinking, which has been so powerful in biology. And we can bring that principle to neural systems. We want to test ideas. We want to test hypotheses, just like a geneticist might, or a developmental biologist. Just as they might knock out a gene or edit a gene with a technology like CRISPR, we want to do the same thing with activity in particular cells. We want to provide it or take it away and see what happens. And we'd like it to be flexible. We'd like it to be fast. We'd like it to operate on the time scale of normal processing in the brain and behavior. And this is what optogenetics has allowed us to do. And by way of analogy, you think about what gain and loss of function has done for developmental biology. This seems like an image out of a nightmare, but it's actually really evocative and powerful. This is a fly, a fruit fly. Normally flies have antennae on their head, but if you do gain and loss of function interventions on single genes that control development of the fruit fly, you can make a leg appear, two legs, a pair of legs appear where the antennae were. And this tells you a lot about what that gene that you altered does. It's involved in pattern formation. It dictates what grows where. And this is the same sort of thing that we want to do and now can do with neuroscience. Now, it's not just to turn the cells on or off. We want to see what happens with behavior. That's the ultimate readout. And so we developed fiber optics to get light deep into the brain to target the cells that we would like to target. And I'll play this movie. This actually was our first indication that optogenetics would work. We did this experiment in 2007. Some of you might know that the right side of the brain directs movement toward and attention toward the left side of the world. And very early on, we wanted to see if this would work in mammals. And so we put in a channel rhodopsin and a fiber optic in the right side of the motor cortex, the part of the surface of the brain that controls movement. And we turned on the light and asked, what's this animal going to do? And so I'll play the movie and you'll see when the light goes on, a little blue dot appear on the head of the animal. And you'll see as the right side of its brain is being optogenetically stimulated, what happens. So immediately the mouse starts turning left, really wants to, passes up the food to keep turning left, circling around, and then we turn off the light right here. Now perfectly happy to stop, look around. Okay, so this is a little eerie, right? It's you suddenly made an animal really want to turn left and then made it stop wanting to turn left. And if you think about what that means, it means a lot. This particular experiment, we didn't learn anything scientific from except that the technology would work. But of course, it's been applied now in thousands of experiments and for thousands of discoveries. One example is one that just a couple years ago that we set up to look at the single cells that are involved in drive conflict, primary drives that we as animals have. We have hunger, we have thirst, we have one set of drives. We also have other drives for social interaction, for thermal regulation, for everything you can imagine. In a part of the brain called the orbitofrontal cortex, there are cells that are involved in both social interaction and feeding, in eating food. In fact, people who have a stroke in this region have something called orbitofrontal syndrome, where they have disrupted feeding and social interaction. So we thought this would be an interesting place to look at how single cells become involved with and adjudicate drives and drive conflicts. And of course, this is relevant to normal decision-making, but also to disease processes. If you think about the eating disorders like anorexia nervosa and bulimia nervosa, these are very fundamentally disorders that start with drive conflicts, social versus feeding drives. And so understanding these cells could help us understand these really seriously debilitating and very mysterious disorders. So we looked into the orbitofrontal cortex of mice. We gave them a little high caloric reward, basically ensures something very, very tasty that we would like to. And we looked at the cells, looked at which cells were glowing with our activity reporter strategy in response to a drop of tasty fluid. And I'll play this movie, and we saw right away that there were some feeding cells that responded to a caloric reward and others that didn't. The feeding cells are F in blue, and you can see those cells responding when the gray vertical bar shows a feeding drop was delivered. Here again, whoop, blue cells go up. They're responding. It's going to happen again right here. Watch the blue circles on the left. You can see them responding. The non-feeding cells are doing their own thing, but they don't care about the feeding. So right away, you've got this very clear principle. Cells doing one thing are intermixed with cells doing something else. And this was one of the fundamental challenges of neuroscience that we struggled with for so long. We could see these correlations, but we didn't know what actually mattered. Why are these cells more active during feeding? Are they doing something about it? Are they causing the feeding? Maybe, or maybe not. Maybe they're just reporting on the feeding, or maybe they're doing something else entirely. Maybe they're there to shut down feeding in response to terminate the feeding bout, for example. We just don't know. All we see are correlations at this point. And here's where optogenetics comes in. This gives us causality. Now we can test what actually matters. And so this is what we did. We delivered one of the Volvox-derived opsin from that spherical colonial algae that we discovered. It's called B. reaches. That's the name of the opsin that came from the Volvox algae. And we guided light to just the feeding cells. And what we found was that each little tick here is a little lick the animal gives for its caloric reward. Here's where the reward becomes available in time: 0 seconds, 5 seconds, 10 seconds. And these are different trials. We do it many times. The animal has very consistent behavior. It licks for a certain amount of time and then stops. That's the normal behavior. Now, if we illuminate to activate the cells optogenetically, lo and behold, what we see is increased and prolonged feeding. So actually activating these cells does matter, and it matters by promoting more feeding. So these are cells that are active and promote and drive feeding, and they're sitting there in orbital frontal cortex. We wouldn't have been able to assess this in any other way because these cells are intermixed with other cells. We can't access them separately with an electrode. They're not genetically different from the other cells. We would have no way of knowing. But this was clear. And by the way, if we left out the opsin and we just used light, there was no effect. That was reassuring. It wasn't some side effect of the optical stimulation. So that was what we call a control experiment to make sure we know what we're talking about. So that was pretty exciting. But now you might say, well, all right, you stimulated some cells, fine, but how do you know that it mattered that it was the feeding cells? What if you stimulated some other cells? Not only that, what if you also stimulated cells that were involved in another primary drive, a very motivating drive like social interaction? What would happen? And so we set up an apparatus where a nice, friendly juvenile mouse, non-threatening, can wander around here and occasionally pop out through here, and they can have a prolonged whisking and sniffing and interactions that are meaningful to them. And what we saw is that there were social cells. There were cells that on different trials here, the yellower and redder colors mean more active cells. And each of these horizontal rows is a single cell and its activity upon the juvenile coming out into this social zone is reported here. And a lot of the cells were social. They responded to the social interaction and not to feeding. Okay, so this was interesting. But again, this is the natural activity of the cells. It doesn't tell us what's causal. But then we could ask what happens if you stimulate the social cells, what happens to feeding? Does it also increase the feeding? By the way, this is an important consideration. Anytime you do an experiment like this, you want to do controls and make sure that when you set up a social interaction, that it really, what matters is that it be social. And a skeptic might say, well, maybe it's just surprise. Maybe what you're looking at is surprise cells or novelty cells. Suddenly there's a mouse there. Maybe it's a shock cell instead of a social cell. And so we set up controls to do this. And here's a fun control that was also informative. We 3D printed another mouse and had it pop up unexpectedly. So it's not a social target, it's just a 3D printed mouse, but it's just as surprising. And if you look at the social cells in this animal, S1, S2, S3, which were very powerfully responding to the true social interaction, you'll see what they do as I play this movie when the 3D printed mouse pops up. And appropriately for Halloween, this is a little scary. Oh, there's the 3D printed mouse popping up suddenly. And amazingly, the social cells did nothing. They don't care. They're not shock or surprise cells. They don't care. There it is again. No response. There are other cells that are novelty related and they did respond, but not the social cells. So we really did find social cells And we were able to study them. And what happens when you stimulate those instead of the feeding cells? Do you get also an increase in feeding? Actually, no. You get a decrease in feeding. You get a social cell-driven suppression of feeding. And that lasts for a few minutes. It eventually is corrected, but for a few minutes, you have this suppression of feeding. And by the way, if you stimulate non-social and non-feeding cells, you get no effect at all. So it matters which cells you pick out. It matters that you can pick them out with guided light. And together, what this means is we can study primary survival drives in animal behavior with causality and do it at this elemental level, the single cell level, this fundamental component level at which the brain operates. So this is an example of what makes this approach so exciting. We can get to this fundamental level in behaving animals. And it's quite remarkable, I think, to realize this, the roots of this go back to a Russian botanist more than 150 years ago, but also more recently in 1971 to just studying ancient forms of bacterial membranes and seeing what happened when they responded to light and solution changes. And I think that's a testament to the importance of true basic science discovery. Now, it's not limited to studying primary drives. Anything that the brain does, you can study with this approach, and even very high level what might have been thought as uniquely human conditions and processes can also be studied with optogenetics. And this is a self-portrait by the American pop artist Andy Warhol, and you can see several forms of himself spatially shifted from each other, retaining some element of the common features, but shifted in space. And this, I think, is an evocative depiction of what we call dissociation in psychiatry. What is dissociation? This is very common, not well understood. It's a state of the brain in which important things are uncoupled, separated from each other, including sensory stimuli, things that happen, awareness of things in the world, from what we call an affective or an emotional response. So it's not like anesthesia or numbing or being unconscious. You're still aware of everything happening. And people describe this who are dissociating, it doesn't matter that it's happening because it's not attributed to themselves, to their self, which is viewed as separate. Now, how interesting is that? That is amazing. Now you might think, well, that sounds kind of wacky. I don't believe it, but it's very consistent across major kinds of human condition described by people who'd never experienced it in astonishingly similar ways. It arises in post-traumatic stress disorder, in the dissociative disorders, in certain personality disorders like borderline. It's caused by drugs, PCP and ketamine. Trauma. In people who've had severe trauma, more than 70% of people will experience and report dissociation. And overall, in the general population, about 10% of us will experience dissociation. And the descriptors are astonishingly common. The separation of being in the passenger seat instead of the driver's seat, still aware of everything going on, or like watching a movie of your life, but without the emotional response because it's not really you, you're watching it, okay? Now, how interesting is that? And is it possible that this could be studied? And it turns out optogenetics was really helpful in understanding this. And the way we approached this was first to study what these altered states might be, and starting in mice. And we carried out what you might call an unbiased screen using optics. We didn't know what we were looking for. We had no idea. How could something like dissociation happen? So we did an unbiased screen. We looked at the whole surface of the brain of an awake, healthy, comfortable mouse. And we administered either a salt solution, which did nothing, just a control solution, or things like ketamine. And we saw something amazing, completely unpredictable, that we certainly hadn't predicted pop out. So again, we're using these fluorescent markers of activity to see what's going on using optics. In the animal, just getting a saline solution where nothing really is happening, we see these waves of activity Red is higher activity, blue is lower. With the dissociative drugs like ketamine, we saw this little patch of the brain that is a part of cortex, the surface coating of the brain, and a part of it called the retrosplenial cortex, RSP. There's a little patch right here, and it was pulsating about 3 times a second, very consistently in a localized way. We definitely did not predict that. That was out of nowhere. What is going on with that? Well, it was only in retrosplenial cortex, not in these other parts of the brain, and only the dissociative drugs like ketamine, PCP, other drugs that were dissociative caused this retrosplenial oscillation. Other drugs, even very potent psychoactive drugs like LSD, a hallucinogen, but not a dissociative agent, didn't cause this. And it was very specific. It was only the cortex of our brain. This covering is like 6 plastic wrap layers on top of each other. And it was really coming from just the 5th of these 6 layers. If we had a label for just layer 5, we saw this oscillation, but if we looked at other layers like layer 2 and 3, we didn't see that oscillation over time. So it was very specific, and that made us think this was a meaningful thing. And so we decided to look at behavior and see if we could separate this stimulus detection from the emotional or affective response to the stimulus. And so we did a safe, well-tolerated test in mice. If they have a brief exposure to something that's too warm, there's a reflexive withdrawal of the paw, just as we would do with our hand. And that means they've detected the stimulus. And then there's a licking response, and that's more prolonged. And you might classify that as an affective or emotional self-protective response. It's beyond just detection. It's taking care of the self, taking care of the body. And what we found, amazingly, was that we looked at different doses of ketamine, the dissociative agent ketamine, and the stimulus detection, this reflexive paw flick, was unchanged. You had the same number of withdrawals at any dose of ketamine where the animal was still conscious. But the affective or emotional response, the self-protective response, completely collapsed at a particular dose, 25 to 50 milligrams per kilogram. And that was the same dose that caused the oscillation. You can see that oscillation appearing at about the same dose. All right, so this dissociative-like behavior appears at the same dose where this oscillation appears. And we know this is happening and it's specific to the dissociative drugs. Still, though, it's correlations. We don't actually know that the oscillation is causing the dissociative behavior. And that's where optogenetics came in. And what we did was effectively used stimulation and inhibition with different microbial options to turn on or off. We had blue ones to drive activity, blue light responsive ones, and yellow light responsive ones to shut down activity. So we could stimulate, inhibit, stimulate, and we could create any oscillation we wanted. And because these are genes, we could target them to even layer 5 of retrosplenial cortex, and we could provide the rhythm to the right cells at the right time, which is what we did. A lot of technical details here, but that's basically what we did. Layer 5 of retrosplenial cortex, we provided the rhythm. And what happened, it didn't affect stimulus detection at all, but it powerfully reduced this self-protective response. And it mattered that you did it in retrosplenial cortex, not in other parts of cortex, like even somatosensory cortex that's involved in stimulus detection didn't have that effect. So we knew this was causally important now. Now, here's where we returned to the human condition, and I think in a very interesting way. Here at Stanford, as many major hospitals have, we have an epilepsy monitoring unit where people who have intractable epilepsy are brought in to record, to listen in, even deep in the brain, to see where the seizure, where the epilepsy is starting. And that's to guide neurosurgeons to go in and take out the tiny part of the brain that's causing the seizure. But you really want to know, if you're going to take out part of the brain to try to cure someone who's really suffering from lifelong intractable epilepsy, you really want to be sure which part you take out. So they do this recording beforehand to detect where the seizure is starting. And we had a patient who came to Stanford who had what's called an aura. People who have migraines might know about auras. It's what comes right before the migraine, where you feel a little different. With epilepsy, you have auras too. Before a seizure, you feel a little different. And this patient had an aura that was dissociative. He dissociated as part of his pre-seizure condition. And it was classic dissociation. He said, I was listening to two parts of my brain speak to each other in a way that a third part of the brain, which I considered to be me, was able to listen. Okay, and so that patient had already come through our center, but we'd already carried out this unbiased brain-wide screen in mice, and we said, hey, we've got these recordings in this person. Let's go see what was going on. And if you look at the recordings, we looked at all of them, we found a rhythmic oscillation during the dissociation as this human being was reporting it. And where was it? It was in the human homologue of the same spot that we saw in the mice, this retrosplenial cortex. In human beings, there's a collection of areas, including retrosplenial cortex, that are in this, what we call the posteromedial area, and that's where the oscillation was appearing. And it was just there. We saw those oscillations during the dissociation in these deep posteromedial areas, but not in other areas. Now, what was even more amazing, that's listening in on the human brain during dissociation. We saw this correlation. What about causation in the human? Well, part of the normal epilepsy mapping process is to also stimulate with the electrodes to see if you can trigger a seizure-like event at a particular spot. Then you really know that's the spot that you may want to take out. And it turned out that stimulating at these spots, the oscillating spots, caused dissociation in the patient. And this beautiful descriptor was from this patient, feeling like the way a pilot can lose control of a plane, like being forced out of the cockpit, but still seeing what's happening, still seeing all the gauges, all the information. Okay, classic dissociation was stimulating at this spot, and it was only the spots that had this rhythm that caused the dissociation as shown here. Other spots didn't have that effect. So now we were on a very firm foundation in understanding that this region and this rhythm could be involved. And so then coming back to the case of a mouse where we can do more experiments and understand even more deeply, we asked, how could this happen? We wanted to come to a deep understanding of what this state that had been so mysterious, not even really believed by people, but the doctors knew, the physicians knew, and the patients knew. They knew it was real. And if we can get it to biology, think how important that is for understanding, for acceptance, for empathy. And this next step really brought us to that mechanistic level of understanding. And we did that by thinking about how retrosplenial cortex was wired up. And we did the equivalent in the mouse, what we'd done in the human, of listening in across the brain. Okay, now I've talked a lot about the cortex, the surface part of the brain. There are, of course, deeper parts of the brain too. And there's a deep part of the brain that does a lot of the routing of information from different regions of cortex, passing them on down to the spinal cord, passing on to other areas of cortex. And that's called the thalamus. It's a deep structure under the surface parts. And so there are all kinds of cortical regions, including retrosplenial cortex, and all kinds of thalamus regions or thalamic regions. Now, it turns out, if you look at the neuroanatomy, you look at the wiring, just as you might look at a circuit wiring diagram of a computer, retrosplenial cortex is connected physically by its wiring to some thalamic regions, but not others. So these two, but not this one called AM, for example. And so we thought, hey, let's see if maybe this oscillation is in training, is bringing these parts of thalamus along, but not this part. So maybe this is effectively creating part of the brain doing one thing and leaving another part of the brain out of the party. So we did the equivalent of putting in electrodes to listen in all across the brain. And this is a cool plot. This is a cool way of looking at the data called a correlation matrix. So we put all the areas of the brain. It doesn't really matter what they are, just letters right now, but you can see retrosplenial cortex here. And we also put them along the x-axis and the y-axis. And before ketamine, which is on the left, everything is weakly positively correlated. More red means more positively correlated. Blue is negatively correlated or out of sync. And in the normal state, there's a lot of weak positive correlations. Now you put on ketamine, And what do you see? Retrosplenial cortex becomes very powerfully correlated with itself. And that's what you'd expect. All the cells, because they're all in this oscillation together, they're going to be very correlated with each other now, much more than they were before. And so that's why you see that little patch of red there. But what's going on here? These are these three thalamic regions, AM, AV, and LD. AV and LD, these are the two that are connected to retrosplenial. And look, they're coming along with it. They're coming along with this oscillation. They're in sync with retrosplenial cortex. Here's their positive correlation with retrosplenial cortex. But this one, AM, that's not wired up with them, look at this, it's become negatively correlated. It's being pushed out of sync. And you can actually look at this in the raw data. Here are the ones that are wired together and they're going in sync. These two thalamic regions are in this magenta color, and you can see them oscillating along with the green retrosplenial cortex. But this one that's not wired up is out of sync and forced to operate on another rhythm in another way, and never able to be active at the same time as the others. So no part of the brain is being shut down. Nothing is becoming numb or unconscious. Everything's still active, but they can't work together anymore. And in fact, if you look at retrosplenial cortex and these parts, the thalamus that it connects to, well, this is only part of the picture. Here's this part that's out in the cold. This part projects and connects to many other parts of cortex. And so you're going to break apart the brain into two systems that are out of sync, still active, but not able to form part of the same whole. And so this was an insight into what dissociation actually may be physically, materially, that optogenetics and an unbiased optical screen provided for us. And so you can see even these very high level concepts, so mysterious and so interesting, are accessible. Now, the Lasker Foundation folks, and I think very much rightly so, as part of this outreach, asked me to talk about projections, the book that I wrote, and that addresses this new understanding of ourselves, our brains, and our dysfunctions, our suffering, that has come through advances in neural technology like optogenetics. And for me, as a psychiatrist, I focus on treatment-resistant depression, people who suffer very severely. I have stories that I share in the book about my own personal experiences and stories of patients. And these human stories serve as branching off points to explore more fully the human experience in health and in disease. And indeed, this book does, in the first chapter, go into grief and depression and loss, but with the perspective that modern neurotechnology like optogenetics brings. And it starts from a story of a patient that I saw who had some of the parts of grief, but not all of them. He'd lost one of the features of grief, which was crying. He had lost the ability to cry, but he was suffering from a horrific bereavement that had happened. And his loss was so great, it could barely be comprehended by me. I was a resident, a psychiatrist in training at the time, and just completed medical school. And it was inexplicable to me how some of the parts of grief could be present and others absent. And the insight that optogenetics later brought us showed us how complex behavioral states like grief, like anxiety, even positive things like parenting or caring for our offspring, are assembled, are pieced together by different components, connections or projections across the brain. And here's an example of how anxiety, that state that we all know too well, some of us, how that is put together, how it's pieced together. And optogenetics gave us that insight, how the parts work together to create the whole. Now, if you think about anxiety, it's got different parts to it. It's got different features. It's got a physical change. There are respiration or breathing rate changes. And then there's also behavioral changes. We start to avoid things that might be risky. And then it also feels bad. We call this subjective preference or subjective valence. So you've got these different parts. It feels bad, it affects our behavior, it causes physiological changes. That's a complex state. You can't just say anxiety, it's got a lot going on. Well, optogenetics help us figure out how that state is put together, how it's assembled. We introduced a channel rhodopsin into one part of the brain, and then we used a fiber optic to deliver light to one or another other regions of the brain, even far across, far distant from where we had introduced the channel rhodopsin gene. And what does that do? That lets us look at the projections, because the cells that live here in this region, they'll start making that channel rhodopsin and they'll ship it down the axon, down the outgoing wire that connects that cell to another cell across the brain. And in that downstream region across the brain, the only thing that responds to light is that outgoing connection that started here and ended here. So by delivering your fiber optic here, you pick out the cell that lives here and sends its connection there. And we found that the cells in this region of the brain called the BNST that project to the area where the dopamine neurons are deep in the midbrain, that governed that feel good or feel bad part, the subjective valence part of the state. Another projection to a region called the lateral hypothalamus affected the behavior, but didn't affect the preference. And yet another region went to the pons, went to the brainstem and affected respiration or breathing, but didn't affect the others. And so this complex behavioral state of anxiety was pieced together by these different projections across the brain. And my friend and colleague, Catherine Dulac, did a similar thing with parenting. And you might think that's a positive state, of course. How could that be broken apart into different components? But it can be. And the two components that were looked at here were caring for the young directly by grooming them. We all know brushing kids' hair, giving them baths, all that. This is part of parenting, very much part of it. And then another part is climbing over a small barrier to retrieve the young, bring them back into the nest. I got 5 kids. I know retrieving, and that's a big part of parenting. Contain them, and if they get out, retrieve them. Okay, that's a big part of it. Turns out the different components are each controlled by different projections. There's a region of the brain that projects from here to here. The name doesn't really matter, but this projection controlled the grooming, the caring for the young, but it didn't affect the going out to retrieve them and bring them back. That part was not affected, but a different connection did the going out and getting the young, but didn't affect the grooming. And so these different parts of parenting are assembled. Our behavioral states, our inner states are put together by projections. And this is another insight that optogenetics helped reveal. The other chapters, as I move toward the end here, I'll just say that there's a chapter that deals with mania as part of bipolar disorder. And one of my other major clinical interests is autism spectrum disease. And there's stories that relate to autism, both elevated and reduced social function. Borderline personality disorder, where dissociative states are very often seen. Schizophrenia or psychosis. Eating disorders, where the sense of self is altered so powerfully, and where deep insights into regions of the brain like the retrosplenial cortex have been so valuable. And then into depression and dementia at the end of life. All stories that touch on my own experiences in human stories, but help, I think, try to bring the insights that modern neuroscience have brought us, including optogenetics, to help us understand ourselves better and those of us who are suffering. If you're Dutch, you can read it in Dutch. It turns out projections doesn't translate too well, and so there had to be a different title. This is Dutch for insights instead of projections. As I found, even in British English, a different title was required. Apparently, projections carried too many negative connotations in the United Kingdom, and so the title there is connection. So we even had to translate it into British English. And the Spanish folks liked connection, so it's conexiones in Spain. But all of this is here I'm so grateful to the Lasker Foundation for all the work they do in helping reach out and communicate science. And this is my effort also to help everybody understand what's been happening in the science and to help understand themselves and each other more deeply. We have a long way to go, of course. There's every window we open up creates new questions, new problems, new challenges, but we can now start to look at how our uplifting states and our suffering arise from connections, projections, components, elements that we can study now with precision. And to close with, it's just, I think, also remarkable, again, I said this earlier, but worth underscoring how the basic science understanding, getting to the atomic level understanding of how microbial proteins work and doing purely curiosity-driven science has taken us to this point in human history that could never have been predicted. Again, I'm so grateful to the Lasker Foundation, to all of you for spending your time with me, and for all my amazing students and postdoctoral fellows, colleagues and collaborators around the world. And I'll wrap up there. Thank you.
Thank you.